Okay, well, in that case, this is that sounds like the perfect cue to suddenly switch roles and be today's chair for the UCL Catalyst Seminars for Children and Young People's Mental Health. So, welcome everybody. It's really wonderful to see so many of you here today, and we're very excited. My name is Pasco Fearon, and for those of you who don't know me, I'm a professor in the clinical educational health psychology department here, and part of the Children and Young People's Mental Health implementation group that set up the Catalyst seminars, really distinctly not capitalist, because they're free events. Back in at the very beginning of 2021, so it's a great pleasure to welcome you all today. I'm really excited about today's session. It really is actually, in terms of my own scientific interests, a bit of a dream come true, actually, that this has happened, and I'll come to that in a minute. But I just wanted to, before we start, just say a few words about the Catalyst seminars. I think many of you have now been, but we've this is, I believe, the third in the series, launched at the beginning of the year, and it's really, as many of you will know who are at UCL or who know about UCL, it's an enormous and complex entity, UCL, with a huge amount of incredible research going on. And often, the challenge has been to try to link people together who have common interests but may not even know about each other, or will be working from very different disciplinary models. And we realized some time ago that there was huge untapped potential to stimulate much more interdisciplinary research and build interdisciplinary research collaborations and think about strategic funding awards and so on, if we could link some of these more disparate parts of, or more distant parts of UCL's incredible depth in research in, broadly speaking, developmental science relevant to the mental health of young people. And it's been really successful already. We've got lots of people coming from UCL and also outside UCL. So welcome to all of you from UCL and outside. It's great to see some of you here. And I think today's seminar is in many ways a perfect kind of exemplar of the ambition that was at the heart of this of the Catalyst seminar, because we're seminar series, because we're hearing from two speakers today that are focused on very similar problems that are absolutely central to the etiology of mental health disorders in children and young people, which is, of course, parenting. And they're studying the underlying neurobiology of parenting, but at very different levels of organization. So Johnny Kohl, who's going to be speaking first, will be talking very much about at the level of neural circuits of behavior of parenting using rodent models. And then Helena, who's doing systems neuroscience with in parents from our species. And so, and of course, there's a lot of work going on more broadly within UCL, looking at parenting as a construct, developing parenting-focused interventions, looking at the epidemiology of parenting. So today is a great opportunity to think about what are the cross-level interactions that we could think about from the level of cells and neural circuits right up to system-level brain systems that could help us to understand how parenting works, its normative development, but also critically how it can go wrong, and what are the sort of underpinning roots of harsh or unresponsive parenting, or of course of maltreatment, given that we know that these are some of the major determining factors in children's mental health, and also some of the major levers we have to pull for promoting resilience within families to ensure that mental health problems don't arise in the first place. So really looking forward to today's talks. And first up is, as I said, Johnny Kohl, who's in the UCL's Division of Biosciences. And Johnny's a group leader at the Crick Institute, which you can see behind him. And he heads up the state-dependent neural processing lab there, which is focusing on how internal physiological states shape information processing in the brain. And a key theme of the work that Johnny's lab does is to look at instinctive behaviors like mating and parenting, and to understand how that is these evolutionarily shaped neural systems can be understood at the level of circuits. Johnny studied biochemistry originally, and preclinical medicine and neuroscience in Germany. Then he came to the UK, to the MRC Molecular Biology Lab in Cambridge. And then he moved to Harvard in 2014. I think that's when his work on neural circuits of parenting behavior really took off. And then in 2019 came back to the UK to lead up his group at the Crick Institute. And his talk's going to be entitled Circuits for Care, Neural Control of Parental Behavior. So Johnny, thanks so much for coming along today. We're really excited to hear your talk, and I'm going to pass over the podium to you.
Thanks a lot, Pascal, for this nice introduction. Thanks to you and Maya as well for organizing this today's series, and to Andrew especially for inviting me to this in the first place. I'm going to start sharing, and you should be able to see my screen right now. Is that okay? Good. Thank you. Okay, so as Pascal mentioned, my lab is interested in how the brain controls instinctive behaviors. So those are the behaviors that animals can perform with very little or no prior experience. And I would argue that some of the most interesting instinctive behaviors occur when animals meet other members of their own species. And during such encounters, behaving appropriately is a matter of great importance and a matter of life and death in some instances. And in some cases, your survival really depends on your ability to elicit a stereotyped behavioral response in others. These mouse pups, for instance, will only live if they can elicit a stereotyped parental response from an adult caregiver. And this parental behavior is absolutely crucial in many species, and especially in mammals such as ourselves, where quite elaborate parental care is one of our defining features. So in mice, because we're only talking about mice in my lab, in mice, parenting consists of a few quite stereotyped motor responses. For instance, here you see a pup retrieval to the nest, which is performed by this mouse father here. And then other motor routines of mouse parenting are nest building, for instance, pup grooming, pup licking, and in females especially crouching and nursing, of course. Now, in addition to these motor routines, parenting is defined by by an increased motivation to interact with infant stimuli in the first place. It's defined by distinct hormonal states, and often or typically by a temporary suppression or downregulation of competing non-parental behaviors. So we have we have stereotype components, but at the same time a very complex, multifaceted behavior. So what do we actually know about the neural control of parenting? Now, it turns out that through lesion and pharmacological stimulation experiments over the last few decades, many brain areas have been implicated in the control of parenting. But one area that has been consistently found to be critical in parenting is the so-called medial preoptic area, or MPOA, as I'm going to be calling it, which is shown down here in this coronal or side section through the mouse brain. So it's this area is sitting at the anterior end of the hypothalamus. And a few years ago, the Dulac lab at Harvard wondered which of these MPOA neurons are actually activated during parental behavior. And what Herbert Wu, a grad student in the lab, did, he used the immediate early gene CFOS, which is an indirect molecular readout of neuronal activation, to show that these MPOA neurons are indeed highly activated after parenting in both mothers and fathers. So you can see these CFOS positive neurons here. In contrast, these neurons were not activated or very weakly activated in virgin male mice, which are normally infanticidal, so they attack and kill pups. Now, so that showed that the MPOA is activated after parental behavior, but importantly, this brain area, like most brain areas, is involved in many other behaviors and physiological states. And therefore, Herbert was wondering whether these parenting active neurons share a distinct molecular signature. And indeed, after a prolonged screening effort, he found that the neuropeptide galanin is enriched in those neurons activated during parenting. And he could now use this, this information to use mouse genetics to specifically manipulate the state of these neurons. And intriguingly, what he found is when he ablated these neurons, this manipulation abolished parental behavior in both males and females. And even more intriguing, when he artificially activated these neurons, in otherwise non-parental virgin males, he could elicit aspects of parental behavior in these mice. So these so-called MPOA galanin neurons are therefore critical for parenting in both males and females. And that was a pretty substantial contribution to our understanding of how parenting gets regulated in the mouse brain. However, it also left a pretty substantial question open, namely, how can such a small population of merely 10,000 neurons out of about 100 million in the mouse brain, how can such a small population actually control or orchestrate such a highly complex behavior? And that's where I, as a postdoc, hypothesized, maybe unsurprisingly as a circuit neuroscientist, that these neurons might achieve this by assuming a hub or a central position in a brain-wide circuit dedicated to the control of parental behavior. And my first goal became to anatomically outline or define this hypothetical circuit. So I started by using viral tracing techniques, in this case, monosynaptic rabies tracing, to directly visualize the monosynaptic or direct inputs that these neurons receive. And what I found was quite striking. These neurons seem to receive inputs from many brain areas, some of which are shown here. And when we visualize these, all of these input brain areas in this map here, so here's a sideways view onto the mouse brain. The left is anterior, right is posterior, and each of those dots represents an area that provides direct input into MPO galanin neurons. We found that these neurons receive inputs from modulatory areas, so those areas that broadcast the animal's physiological state. They receive inputs from the reward system and from pheromone processing pathways, for instance, which is interesting because pup-released pheromones are critical parenting cues in mice. So these neurons therefore receive inputs that convey both external and internal modulatory signals. And the question now became, how do they integrate these signals, and how do they then use this information to orchestrate this behavioral output of parenting? So again, to visualize the projections of these neurons, I used a viral strategy. I infected these neurons with viruses that allowed us to trace their projections and to visualize their presynaptic sites. And again, what we found was that these neurons project to about 20 or so brain areas, from many of which they also received direct inputs. So there's a high degree of reciprocal connectivity going on in this circuit. And we then did a lot more anatomical experiments that I'll just summarize quickly to understand how these inputs and outputs are organized. And briefly, what we found was that a motif in which we have these broad input combinations converging onto projection-defined subpopulations of MPOA galanin neurons. Now the next question was, okay, this is the circuit organization in the anatomical sense, but which of those 20-odd neuronal projections actually contribute to the control of parenting? And in order to approach this question, we again used a viral strategy. We infected each of those projections and then used CFOS again as a molecular readout of neuronal activation after parenting, and then quantified the percentage of neurons in each of those projections that was activated during parenting. And that allowed us to really home in on 3 of those projections for further functional investigations, namely those to the so-called periaqueductal grain or PAG, those to the ventral tegmental area or VTA, and those to the medial amygdala or MEA. And our next goal basically became to understand how activity dynamics in these 3 projections play out when the animal is engaged in parental behavior. And for this purpose, we needed a method, an activity recording method with a higher temporal resolution. And the one that we chose to use is fiber photometry, which is basically an imaging method that allows to record population activity from genetically defined neurons in behaving animals. And in the next video, you will see an example of such a recording of a mother engaged in different behavioral interactions. So here's a top view of a mother in her cage. I'm not sure if you can see this. So here's the mother. And you will first see her interact with this roughly pup-sized and shaped cracker, so a food item. She will then interact with these caged pups. And then finally, she will interact with these freely accessible pups in the upper left corner. And you can please focus on what happens to the MPOA galanin population activity down here. the line indicates where we are in time currently. Okay, so what you probably saw is was that the line was completely flat when she was interacting with this food item. There was a small increase in activity when she interacted with these inaccessible pups, and then the signal increased quite dramatically when she started interacting with these freely accessible pups. So when we did these recordings in mothers, in virgin females, and in males, we found that MPO-galanin neurons were activated during pup sniffing, during pup grooming, during pup retrieval to the nest, during entering of the nest, and in mothers even during entering of the empty nest. So essentially, these neurons are activated during all active pup-directed aspects of parental care. Now, in contrast, when we selectively recorded or imaged from individual projections, we found that PRG projections were selectively active during pup grooming. VTA projections were weakly active during entering of the nest. And in contrast to this, again, the medial amygdala projections were essentially active during all aspects of parental care again. So this really suggested that these projections are differentially activated during different aspects of parental care. The question now became whether this differential activation also meant that these different projections control different aspects of parental care as well. And in order to address this directly, we optogenetically stimulated and inhibited each of those projections during pup-directed and other social interactions. What we found for these grooming-activated PHE projections, we found that when we activated this projection in otherwise infanticidal males, a significant proportion of those males became parental in response. And at closer inspection, this manipulation elicited pup grooming in both males and females. And conversely, selectively inhibiting this projection inhibited pup grooming in both males and females. Now, when we activated VTA projections, we did not find any effect on pup-directed behaviors directly. But the VTA has a very prominent role in reward and motivational processes. And as I mentioned, an increased motivation to interact with pup stimuli is one of the hallmarks of parental animals. And you can see this here in this mother who is separated from her pups here in the foreground by this plastic barrier. She will go to great lengths to retrieve these pups. We used a similar assay to assess motivational components of parenting, and found that when we, in this assay, activated VTA projections, that led to a drastically increased border crossing frequency in this assay that was independent of a general effect on locomotion. And conversely, silencing VTA projections led to a selective reduction in border or barrier crossing frequency. Now, lastly, we also optogenetically activated medial amygdala projections, but here we did not find any effect on either the motivation to interact with pups or direct components of pup interactions. But in control experiments, namely interactions with an adult intruder, we found that this activation led to a reduction in male-male aggression, and in females, a reduction in social investigation targeted towards a male or female intruder. So we believe that basically, rather than directly contributing to parental behavior, that this projection downregulates non-parental interactions and thereby indirectly promotes parental interactions. So overall, then, this work really suggested a circuit motif in which we have these broad input combinations converging onto projection-defined subpopulations of MPO-galanin neurons that each control distinct aspects of parental care. So it's really a relatively simple model for how a small population of neurons can orchestrate a complex social behavior. In my own lab, we now continue this work into the form and function of these parenting circuits, but a big part of the lab now is really devoted to understanding how physiological states change the form and function of such instinctive behavior circuits, because it is quite well known that having a specific circuits for a specific behavior and being exposed to the sensory environment that normally leads or promotes this behavior doesn't necessarily mean that an animal will engage in this behavior. This is hugely depends on the animal's current internal state. And that's illustrated maybe by these two females here, which are exposed to novel pups in their cage environment. So the female on the left, as you can see, shows some interest in this novel social stimulus, but you can also see that she hasn't built a very elaborate nest, for instance, and she quickly loses interest. Now, in contrast, the female on the right engages in a short period of chemo investigation of these pups, and then quite quickly proceeds to retrieve these pups to her comparatively elaborate nest here. Now, intriguingly, these are one and the same female. On the left side, this is the female in the virgin state. On the right side, this is her after having become a mother herself. So what we see is there's a drastic modulation in how these females react to pups after having undergone pregnancy and now being mothers. So our hypothesis, and that is backed up by a few other observations that I will introduce in the next few slides, is that the hormonal milieu of pregnancy now somehow reshapes these parenting circuits to promote adaptive behavioral changes towards pups. And the way we approach this, so we first want to find out when these changes happen, where these changes happen in the brain, and what exactly changes behaviorally. So in order to approach this question, we simply track animal behavior throughout the course of pregnancy and beyond in two groups. One group is allowed to become pregnant and to wean their pups, and then there's a virgin female control group, and we also have an overreactomized control group to get rid of any hormonal influences at all. And what we see in a nutshell is that that there are differences between these two groups quite early in pregnancy already. In the amount of time that they spend grooming pups, for instance. Then towards the middle of pregnancy, day 12 here, there are differences in how much they engage in chemo investigation. So the pregnant group, for instance, engages less in chemo investigation and proceeds to parental behavior more quickly, and also has built a more elaborate nest, which is reflected in a lower propensity to build new nests during the observation period. But the most striking changes occur in late pregnancy, so day 18 in mice. So here the total parenting time goes up in the pregnant group only. They spend more time in the nest with pups, and a larger proportion of these animals retrieves pup to the nest in the first place, and does so with a shorter latency. So these pregnancy-induced changes, they seem to occur at characteristic time points throughout pregnancy, and they seem to affect specific aspects of parental behavior. And as I said, our hypothesis is that it's the hormonal milieu of pregnancy that underlies these changes. So these very well established and characterized so-called pregnancy hormones, progesterone, estrogen, prolactin, oxytocin, for instance. And in humans, there's also a lot of good evidence that this putative remodeling occurs and is in fact very long-lasting. For instance, there's this recent, I guess, yeah, still recent imaging study that found that there's this quite drastic volumetric remodeling occurring in women after pregnancy, and that is in fact quite long-lasting and was still present until at least 2 years after having given birth. So we believe that the same or equivalent thing happens in mice to remodel these parenting circuits. And our goal is to find out where in the brain this actually happens. So to this end, we are doing a few complementary, using a few complementary approaches. One is to simply ask which parts of these circuits are actually responsive to these pregnancy hormones. And to this end, we are assembling these brain-wide multiplexed in situ hybridization atlases, where we visualize, where we can address which individual neurons express receptors for all of these major pregnancy hormones. And that will allow us to then identify target regions for more functional experiments. We also combine this with structural MRI, so in mice this time. So what we do is we take ex vivo scans from mouse brains at different stages of pregnancy. We then make a average brain out of all of these samples, and then register each of our samples to this template brain. And the deformation that each of those brains now has to undergo to be matched to the template brain gives us a readout of how much volumetric remodeling occurred between the one sample and the template. By directly comparing, for instance, the virgin group with the late pregnancy group, using this so-called deformation-based morphometry approach allows us to identify where volumetric changes occur between these two groups. And our results, our preliminary results seem to suggest, so here's a comparison between virgin and late pregnancy, for instance, that we have shrinkage of gray matter, especially in cortex, whereas the most drastic increases, the volumetric increases seem to occur in the hypothalamus. And especially our favorite area is also included in that list. So the MPOA down here seems to be increased in its volume. And that's quite intriguing because we also have other data suggesting there's interesting things happening in the MPOA. We know that many of the neurons located there are responsive or have expressed the receptors for these pregnancy hormones. And my previous work showed that the population activity of these neurons during pup interactions is increased in mothers as compared to virgins. So to now figure out how whether the state or whether the form and function of these neurons is somehow remodeled during pregnancy, Rashida in the lab, a postdoc, is performing patch clamp recordings from brain slices of animals at different stages of pregnancy. And what she's already finding is that there are quite a few interesting changes. So these neurons, their resting membrane potential is decreased. So they are more hyperpolarized, and in a way that there's in a way less excitable in principle. Although that we can discuss what that actually could mean. Then the membrane capacitance is increased, which either suggests changes to the ion channels that these neurons express, or to the total surface area that these neurons exhibit. Another interesting observation is that these neurons seem to receive more synaptic inputs in late pregnancy. And that is a finding that is really mirrored in the morphological changes that these neurons undergo. So what Rashida finds is that these neurons have more dendritic spines in late pregnancy as compared to virgin, and that is where the presynaptic excitatory inputs, of course, arrive on these neurons. So at the moment, we are trying to figure out which of these inputs into these neurons are remodeled and what the functional consequences are to the animal's behavior and the function of the parenting circuit. So by linking these cellular changes to the animal behavior via the circuit level, we really hope to establish, we really hope to understand how the hormonal milieu of pregnancy can remodel the brain to give rise to adaptive behavioral changes. I'm going to stop here and want to thank again the organizers for organizing this really nice mini symposium or mini event. And I want to thank everyone who was involved in the previous work at Harvard, but especially also my current team at the Crick for their excellent work, especially under these very challenging conditions. So yeah, thanks for your attention. Very much looking forward to Helena's talk and to the discussion session.
Fantastic. Thank you, Johnny. What a wonderful, fascinating talk. So we've got some time for some questions. I thought what we'd do is have a sort of brief period for questions now about Johnny's talk, and then we'll do the same after Helena's, and then we'll have a bit of time at the end where we can talk more generally across both talks and think about linkages and so on. So if people have focal questions for Johnny, please do raise your hand, or you can probably safely jump right in.
Can I ask a quick question?
Please do.
So as a non-mouse person, I mean, this looks extremely elegant, and it's beautiful how you can isolate these different influences on parenting behavior. I couldn't help but wonder what the potential implications might be for humans in situations, for instance, where there might be difficulty in parental bonding. So let's say if there's postnatal depression, for instance, which we know can affect the parental bonding, or in situations where you may have an adoptive parent who has some difficulty in bonding with the child, especially if the child is challenging. So I'm just wondering, have you discussed with human researchers, or what are your thoughts?
So I only sporadically. I mean, that's I guess a very intriguing question. I guess in humans, I can only speculate that things are a lot more complex. There's many more effects, developmental, social, experiential, that will influence the state of these systems, of these neural systems. And I guess Helena will know more about that. There are not that many good models, rodent models, for instance, to model postpartum depression, for instance. There are some genetic models that seem to abandon their pups, but it's unclear what that actually means. That might just be, a general disruption of social bonding or attachment. So the short answer is we don't really have any good models to investigate that. Yeah.
But I guess, I'm not just, I'm postnatal depression was the first thing that came to my mind, but of course there are human, just individual differences in how easily people bond. And you could imagine perhaps a situation where you might use something chemical to kickstart a process if it's contextualized in a broader sort of behavioral therapeutic context, which is kind of not unusual for kind of other human conditions. Sometimes, for depression, for instance, you may need some medication, but often accompanied by psychological therapy. So I don't know. I just think it's potentially has. I'm not saying it's ready to go in any way, but there are potentially interesting directions.
I would agree. No, this is largely speculative, I have to say, but I think there's a good chance, because even in mice, which are, presumably they're always portrayed as, relatively simple animals, with these strong instinctive behaviors. But even we see that it's somewhat probabilistic. So depending on how the mouse grows up, which other litters it is exposed to, it is more or less likely to exhibit spontaneous parental behavior as a virgin, for instance. So that's really there's a lot of randomness there as well. So that suggests to me that you can flip one to the other potentially in the adult. That's one project in the lab, is to understand what the circuit level differences are between animals that are spontaneously parental and those that aren't. Once they are pregnant, especially late pregnancy, they are 100% parental. So that's quite clear. But why this variability exists in the first place, and what the circuit nodes are that mostly contribute to this variability, if there are any specific ones. I think that's an intriguing question. Yeah.
Thank you. Thanks for a great talk.
Oh, thank you.
Thank you, Essie. Really helpful question. I have one question from Huma, who's one of our great PhD students in mental health science. And so she's asking, she's read that women's brains get smaller after pregnancy, thought to be a response to their priority to look after infant survival. Is this how does that link to the volume increases that you mentioned?
Yes, that's a good point. So that brief excerpt, that figure that I showed from that human imaging study, that really impressive study from 2017, indeed showed that all of the volumetric changes were decreases. There was not a single area that seemed to be increased. We find increases in both decreases, which in a way makes sense to me, because the overall brain volume presumably stays quite constant, I would think, I would hope. But importantly, we don't actually know what that means. We don't know what a volumetric change actually means. And that's actually, that seems to be something that a lot of studies simply ignore, and that I find quite curious. What does it actually mean? Do you have more neurons? Probably not. Do you have more processes? Do you have more blood vessels? Do you have more water? I couldn't find any answers. So using our MRI data, that's one of the things that we are planning on investigating. What actually happens there? And that's it's very intriguing. It might even be that when an area shrinks, it becomes more efficient in information processing. It's pure speculation at this point. But basically just saying, okay, an animal, a brain area shrinks, and therefore, the animal becomes more stupid in the aspect, in the behavioral aspect that this brain area controls, is probably not the case. Yeah, exactly. As you say, it could be more efficient, but at this point, we know embarrassingly little how these volumetric changes are actually related to cellular level changes. Yeah.
Great. Johnny, I have a quick question, if that's, well, I hope it's quick, which is, I'm quite interested in human studies of adoption, and that gets kind of interesting in terms of how parental behavior might be cued as well as shaped hormonally. So I just wondered, from what the work you've been doing so far, about the extent to which one sort of can tease apart cue-driven changes in the brain versus hormonal changes.
Yes, I think that's a key question, and there's a lot of classical literature from rats, from Rosenblatt in the 70s already showing that rats, which are normally female rats, are normally avoidant of infants, but repeated exposure can actually turn them into into parental animals, and we see the same in mice, and that's why we have this control group that repeated exposure can kickstart some level of parental behavior. The parental behavior is never as good as that of a mother. It seems to be that you need both exposure and hormones to get the optimal level. And we don't know whether this experience factors impinge on the same circuit substrates as the hormones. And that's actually a pretty pressing question in the lab right now. How can we segregate those two effects?
Yeah, brilliant.
But there's these things seem to have similar effects, experience and hormonal changes, although reaching different plateaus. Yeah.
And I assume that people would then immediately be wondering about fathers and paternal caregiving and how that relates.
So I can only tell you what we found in mice. So what we found was that the circuits seem to be largely equivalent between the sexes. Yeah? And all of the activation patterns that we found were also very similar between mothers and fathers. There's obviously the difference between virgin males and females, where the virgin females are often spontaneously parental, whereas the virgin male mice, they are infanticidal, but switch to become parental in the 3 weeks following a mating experience. So this so-called post-mating switch, which is under investigation in other labs as well. But by and large, the core circuit seems to be equivalent. That there might be more subtle differences that we can't detect at our resolution. But I was overall surprised to find this.
Yeah. Johnny, thank you so much. Absolutely fascinating, and we could carry on talking at great length, but we better move on, and then we're obviously going to come back to quite a lot of these issues, because I can see Helena nodding a lot as you speak as well. So I know there's going to be a great link up here. So on that note, it's my great pleasure to introduce my colleague Helena Rutherford, who I've known for a long time. Helena, haven't we? So Helena is an assistant professor at the Child Study Centre at Yale University, and which is really one of the great global centres for child psychology and psychiatry. I've just recommending that Johnny and anyone else who gets the chance to go to the Child Study Centre does, because it's a wonderful example of interdisciplinary research, all focused on children's mental health. And Helena is one of the world's leading researchers in investigating, as I said earlier, the neural systems underpinning parental behaviour in humans, and particularly, and it relates so well to what Johnny's just been talking to us about, the changes that take place in the brain during pregnancy and in the transition to parenthood. But crucially, Helena has done a lot of work looking at the developmental origins and sources of individual variation in some of these neural processes in humans, which connects very closely to Essie's point, and about potentially clinical applications. So we're really excited to hear your talk. I should also quick plug that Helena has a very important connection to UCL and has done for many years, which is that she has been a key player in UCL Yale bridge program for research, but also for our key, really, master's program in developmental neuroscience and psychopathology, where students spend a year here at UCL, and then they spend the second year at Yale doing research. And Helena looks after all of those students incredibly well, and has done for many years. And it's a brilliant example of great collaboration between UCL, Yale, and also the Anna Freud Center. Okay, and so Helena, without further ado, over to you. Your talk is entitled "Beneath the Surface of Parenting: Insights from Human Neuroscience," and thank you so much for coming, and we're really looking forward to your talk.
Brilliant. Thanks, Pascal. Thank you for that introduction and the invitation to be here today as well. So I'm just going to get set up here. So as Pascal alluded to, I'm going to speak about more of the human perspective as we think about the parental brain. But I think there's really nice comparisons and points for discussion between what you just heard from Johnny and then the data that I'm going to share as well today. So just then, as a brief overview of what I want to share with you over the next 20 minutes is I'm going to introduce just very broadly the concept of the parental brain. And I've deliberately asterisked the word parental here. As you'll hear, most of the research is actually focused on mothers. So we know somewhat about maternal brain changes during the transition to parenthood, but we know far less about dads. And so we'll talk about that, I think, during the discussion as well. So I'm really going to be focusing on changes in maternal brain structure as well as maternal brain function. And I want to spend an amount of time with you thinking about clinical studies of the maternal brain too. So really leveraging our knowledge of how the brain changes with the transition to motherhood to potentially understand more about the impact of psychopathology on the maternal brain and then subsequently maternal behavior too. And then I'm just going to wrap up briefly at the end. So to begin with this broader question about why would we even begin to study the parental brain, and taking a step back before we get to that point in terms of thinking about why we study parenting in general. And Pascal in his opening remarks really clued us into thinking about that idea is that parents play a significant role in shaping their children's development. So not only their physical development, but also their social emotional development too. And in the spirit and the theme of the Catalyst seminars, recognizing parents as being a unique environmental input on children's development is really critically important. With that in mind, then, why would we go to the lengths of trying to then neuroimage parents and have them come in and do these experimental tasks when we could just observe parents interacting with their children at home or in the lab, or we could ask them to complete surveys or questionnaires? For me, there are two reasons why I'm motivated to study the parental brain specifically and go to these extremes to understand more about parental brain processes. The first is in terms of the identification of mechanisms. So thinking about identifying specific neural circuits or areas of the brain, many of which Johnny clued us into in his prior presentation, they're important to parenting and parenting behavior. But also thinking about the stages of processing. And to unpack that a bit more, when we think about how parents interact with their children, when we're observing them, we see very explicit behavior. But there's a whole series of processes that have unfolded prior to that behavior that neuroimaging can really give us a window into understanding in a way that we might not be able to glean from just observing behavior that way. And in the purpose of identifying these mechanisms, then we can start thinking about those as targeting in terms of intervention work. So thinking about specific areas of the brain or particularly specific stages of processing, we can think about how we can optimize interventions or develop new interventions that particularly target those mechanisms that we've identified neurobiologically as being important to expressions of parenting behavior. So that's the first reason I think it's important to study the parental brain. The second reason then is thinking about its potential utility for outcomes in terms, again, of thinking about intervention approaches. So understanding how we can look at neural circuits and how we can look at these stages of processing in response to the administration of a parenting intervention. And with that in mind, we can think about using neuroimaging techniques to understand the parental brain as being relatively objective in terms of the interpretations we can make about the data itself. That it's incredibly hard to fake a brain scan, that we can use it as an objective neurobiological indicator of potential change, that we can use to complement more of our subjective measures, whether they be interviews or surveys, to really complement our understanding of how a particular intervention may have led to changes in how parenting feel and how parents express their behavior, but also in terms of what the underpinnings of that are. So to use both objective and subjective measures in terms of understanding efficacy of parenting-based interventions. With that in mind, then, I wanted to begin by talking about maternal brain structure and what we've learned so far regarding that. And when I'm talking about maternal brain structure, the studies that I'm going to share with you have looked at structure through using MRI scanning or magnetic resonance imaging. So I just wanted to make sure that we're all on the same page here. So here's just a photograph of an MRI scanner. Scanners that we use in research settings are comparable to the ones that you get in hospitals. And our parents will lie inside the scanner and complete various experimental tasks. And we can take structural images of the brain that way too. So in terms of looking then at maternal brain structure, there are two main measures that have been examined. The first is grey matter that you heard Johnny allude to. And when I'm speaking about grey matter, I'm talking about brain matter containing primarily neuronal bodies. So parts of the neurons or parts of the brain cells that are important to information relating how that neuron is going to work. So this is just an image of an MRI scan there. So you can see the grey matter there in contrast to the white matter. So those kind of more central pieces, which is the connections between the different neurons. So grey matter has been one measure. The other measure I'm going to speak about is cortical thickness, and that's exactly as it describes, is how thick the cortex is. So these are the two measures I'm going to show you in the next slide when we've started to learn more about maternal brain structure. So there's 3 studies I think it's important to be on your radar, and Johnny helpfully had queued up one of those studies in terms of thinking about brain changes and volumetric changes from pre-pregnancy to postnatal. So this was a study of 25 women who were recruited prior to conception. They completed an MRI scan and then came back and did a second MRI scan at around 2.5 months postnatally. And so you can see in the cartoon graphic of the brain here, the areas colored in yellow are areas that showed volumetric decreases from the pre-pregnancy to the 2.5 months postnatally in terms of gray matter volume there. The second study I wanted to share also focuses on grey matter volume 2, but to pick up on, again, Johnny's data in terms of thinking about volumetric decreases as well as increases, this was a study of mothers who were examined and completed MRI scans at 2 to 4 weeks postnatally, and then came back at around 3 to 4 months postnatally. And the areas highlighted in red were areas of showing increasing grey matter volume across this time period too. So in line with what you heard from the preclinical studies, that there's decreases as well as increases in grey matter volume across this perinatal period. And then a word then about cortical thickness. And this study is somewhat different, not only because it's studying cortical thickness, but also the participants that were being investigated here. So this was a group of women who were recruited in their 70s. So the study was designed to look at the impact of motherhood later on in the developmental period. And so in this particular study, these women evidenced areas indicated in blue here that showed decreases in cortical thickness, but specifically correlating with the number of children that they had. So the more children these women had, the less cortical thickness they had in these specific areas I highlighted here. And importantly, when comparisons were made between these mothers and a group of non-mothers also in their 70s, there were also differences in terms of cortical thickness. There was decreased cortical thickness in the mothers as compared to the non-mothers, suggesting that not only do we see these short-term potential evidence of neuroplasticity in the maternal brain through grey matter volume, but there's also potentially a lasting imprint on the maternal brain too in terms of cortical thickness. Then just to summarize those initial studies, what we really learned is that there is evidence of plasticity in the structure of the maternal brain as indicated by both these increases as well as these decreases in grey matter volume, but also there's potentially a lasting trace of motherhood in the brain as indicated by these changes in cortical thickness. And I think this mirrors really nicely what we just heard from Johnny in terms of the data from his rodent studies too. So I want to then transition into thinking about maternal brain function. And to really get into that, the question you should be asking is, well, how do we measure something as complex as parenting in the brain to even begin with? That it's such a, it's a verb, it's a doing word. How do we even get into that in terms of looking at that in neural networks in human models? So the way that we've come about this is to think about different parenting scenarios and situations that parents find themselves in that we can then start thinking about in terms of modeling neurally. So this is a very typical situation for parents that this is our infant Amelia, who's very happily playing with her in her jumperoo with this farmyard animal set. And she pressed a button and inadvertently that cow figure just popped up. So she was completely unexpected that cow to appear. And we know that sudden abrupt onsets for infants can be quite distressing and dysregulating. And so you can see here Amelia's response. She gets upset, she gets distressed. That cues parent here to then elicit nurturing or caregiving behavior. So mum or dad might step in, pick up baby, try and soothe her, and ultimately try and help her regulate her emotions so that she can go back to playing with her farmyard animal set. So a very typical scenario, baby gets distressed, parent steps in to try and alleviate that source of distress. So we can think about this then in terms of stages of processes. And so this brings in the idea that I mentioned at the beginning is in terms of identifying mechanisms and why we would use parental brain research here to look at mechanisms that might underscore parenting behavior. So we can think about the infant signal here. So that's Amelia's initial cry, that cue of distress that's signaling to parents, something's wrong, something's upset me. And so parents, they might be in the same room, they may be in a different room, but they detect that infant signal. They hear Amelia's cry and they'll come towards her in order to be able to see what's happening. But parents are having their own response to hearing that cry too. They may be having an emotional response that they're trying to regulate. They may be trying to start to interpret what baby needs. And ultimately, this cascade of cognitive and affective processes leads leads to the execution of a parental behaviour. And the cue, or the purpose of that behaviour then, is to modulate the infant signal, so to decrease the infant cries in this particular context. So in essence, what we're doing when we're trying to image parenting or use our neuroimaging methods to measure parenting, we're trying to look at these different stages of processing to understand what the neurocorrelates of those are and how they may relate specifically to parenting, to get that mechanistic insight that we think is so important, particularly as we start to think about intervention approaches. The vast majority of literature is at this very initial stage, thinking about how parents detect infant signals, how parents look at infant faces and listen to infant cries, and what the very neuroactivity is in response to that. So when we think about maternal brain function, there are two techniques that I'm going to share with you data from that we started to learn and understand about what maternal brain function looks like using the stages of process approach. The first is functional magnetic resonance imaging. So similar to what we discussed before, that we have mothers and fathers going to the scanner and we look at neurocorrelates this way. This time, in addition to looking at structural brain images, we also show them photographs of infant faces. It might be their own baby, it may be an unfamiliar baby to them. Baby may be smiling, they may be in distress, but the idea is to look at the neural responses to those very salient infant signals. And then we get these very elegant images here. So this is just a sagittal slice through the brain where you can see very clearly structural information, but the clouds of color here, so the yellows and the reds and the blues, are where we can understand the functional information from. So identifying specific regions of the brain that are responsive to how parents are perceiving infant signals of emotion. So that's our fMRI technique. The second technique is called electroencephalography, or EEG, and specifically ERPs, or the event-related potential technique. So EEG works very differently to MRI. What we're doing in this particular technique is we have parents wear a surface scalp net of electrodes, and we're able to non-invasively measure brain activity from the surface of the cortex. We average that together to look then at neuroactivity across a series of experimental conditions. Like fMRI, we also show photographs of infant faces or have parents listen to infant cries, and our data looks very different. So unfortunately, it's not quite as intuitive as fMRI data, but just to walk you through what this looks like, on the horizontal axis is time in milliseconds, and the vertical axis is the size of the neural response. And as ERP researchers, we get very excited about the different brainwaves, so the peaks here that you can see that I've circled in blue, not only how big they are, but also about how quickly they happen too. And so we use both of these techniques because fMRI tells us where in the brain and gives us that neural circuitry information, but ERPs really tell us when in the brain and the size of that neural response, and gives us that temporal information that's really important to understand too. Those are our techniques then. So I wanted to share with you a little bit about what we have learned. And I've deliberately leveraged knowledge that is going to be important when we think about psychopathology. And so I really focused here on specific neural circuits that have come up time and time again when we think about the impact of clinical disorders on the maternal brain too. There's a lot I could cover, but I've tried to really rein my enthusiasm here. So the first study I wanted to share with you was from Lane Chauvin's group, where he was looking at how mothers perceived infant faces. And the key take home to say from the outset is that infant faces, not surprisingly, activate areas of the brain that are implicated in how we process reward. For the parents in the audience, and also for the non-parents, this is very intuitive and perhaps not surprising, but it took us until 2008 to have a series of studies in place that were reliably showing the specific response. And it becomes important, as I alluded to, in terms of thinking about psychopathology. But here you're looking at a study where mothers viewed photographs of their own baby matched with a demographically matched unknown baby, showing positive, negative, and neutral expressions. So mothers were in the scanner, viewed those images, and what you're looking at here is the take-home findings from the paper in terms of different neural regions that are implicated in these maternal brain responses. I'm not going to go into this in too much detail, except to say that the areas highlighted in yellow are areas that show differences in the maternal brain when mothers viewed their own baby as compared to an unknown baby. But importantly, in green are when mothers were viewing own happy baby faces compared to an unknown happy baby face, that those areas implicated in green are very important in terms of reward processing. And this is a point we're going to come back to later when we think about the salience of when mothers are looking at their own baby smiling. So very intuitive, but also very important findings to get us going. We've also learned that infant cries activate areas of the brain implicated in reward processing too. And at first blush, that might seem somewhat counterintuitive in terms of, well, does that mean that parents are finding infant cries rewarding? And we don't think that that's the case here. And to pick up on a point that Johnny made earlier, it's around, we think this reflects motivational processes. So that when infants are crying, that clues in a need to respond. That reward is not only about the pleasure that we get from a specific event, but also our drive to seek out that event too. And so we think in the case of seeing that these areas of the brain are implicated in reward processing, that you can see here in terms of the nucleus accumbens, the caudate, the orbital frontal cortex, that these areas are around motivational drive as much as the receipt of reward and experiencing of something pleasure, that there's an inherent need to think about reward beyond just this pleasurable experience that we have, but also in terms of how it motivates parenting behavior as well. We've also learned that maternal brain responses correlates with different aspects of parenting behavior too. So this is really critical, that we can have parents come into the lab, we can do our ERP studies and our MRI studies, but can we validate them? Do they have any utility beyond just looking at what images of the brain are responsive or what brainwaves we can see as a consequence? So a number of studies now have documented that maternal brain associations between are there for how mothers bond with their infant, but also in terms of how they perceive their infant as well. We also know that maternal brain activity correlates with how sensitive they are in their behavior towards their children during free play interactions. And we've seen in our own work how maternal brain responses also are correlated with aspects of mentalization, or the mother's capacity to think about their child as a psychological agent. So really important to link brain as well as to cognition and behavior when we think about these different stages of processing and the utility of using neuroimaging to study the maternal brain. One of the focuses in our lab is to be thinking about, can we use our maternal brain data to predict outcomes, so outcomes for parents as well as outcomes for children. So to push this idea one step further of moving beyond cross-sectional studies and looking at the associations between brain and behavior, but then to start looking at predictions and thinking about using brain imaging data to predict outcomes. So we've done this in a couple of different ways. So one way we've looked at this is in terms of using fMRI and looking at neural networks that are implicated in how mothers process infant faces and infant cries. So in this particular study, we looked at mothers at 2 months postpartum and then again at 8 months postpartum. And we had mothers complete an assessment of how anxious they felt in terms of forming a bonding relationship with their child. And we were able to reliably predict changes in neural strength of our networks identified as they were viewing infant faces and listening to infant cries. We were able to predict changing levels of maternal anxiety in the way that they formed a bond with their child across the postnatal period. So suggesting, yes, that there is an opportunity here to look at prediction. We've also gone one step further to think about, well, can we measure maternal brain responses in pregnancy and predict outcomes postnatally? So we use ERPs as our main technique in pregnancy as a way of looking at neural responses to infant faces and infant cries. And in this particular study, we recruited a cohort of women in their 3rd trimester of pregnancy, had them view infant faces, and then invited them back at 3 months postpartum to complete an assessment of mentalization. And we were able to predict, based on their neural responses in pregnancy, their levels of mentalizing at 3 months postpartum. So it's exciting to think about the potential perinatal origins of parenting and thinking about potential markers that are there in pregnancy that we can use to then predict outcomes postnatally, because we know parents' capacity to mentalize has significant implications then in terms of children's social emotional development as well. Transitioning then into thinking about maternal psychopathology. And I wanted to put a placeholder here to think about the impact of psychopathology, not only on mothers during pregnancy and postnatally across their child's development, but also prior to conception too. That psychopathology may be there and may be throughout the pregnancy and postnatally, but may also onset during pregnancy or may also onset postnatally. So psychopathology is a factor that we need to think dimensionally across the lifespan and how it might impact maternal brain, but also parenting more generally. So I wanted to revisit the slide I shared with you earlier and thinking about maternal depression or postpartum depression specifically here. So this was a study that was conducted where half the mothers had postpartum depression, the other mothers were healthy controls without depression. And so again, what they did in the study was listen to infant cries. So it was their own baby crying, an unknown baby crying, and they had a third condition, which was a white noise stimulus, just to have a non-infant aversive stimulus as a comparison. So the areas highlighted in yellow are just the main findings for the mothers without depression, listening to the different types of infant cries. But what I want to highlight here is that there were significant differences in how mothers responded to infant cries in the areas implicated in red as a function of depression status. So specifically what these researchers found is that mothers without depression had their greatest response to own baby cry compared to unfamiliar baby cry and then white noise. But the mothers with postpartum depression didn't differentiate in terms of their neural responses to those auditory stimuli. They had a comparable response for their own baby cry, an unknown baby cry, and a white noise. Given what I've already shared about the importance of reward processes and motivational drives as being important cues to drive behavior towards infants, particularly when they're in distress, one interpretation of these findings is that maternal depression may be attenuating or decreasing the motivational salience of these infant signals of affect, as evidenced here. Maternal addiction has also been studied in terms of its impact on how mothers process infant cues. And similarly, it also seems to decrease reward brain responses, similarly to how we just observed in terms of maternal depression, decreasing reward responses as well. So in this initial study I wanted to share, this was a group of mothers who were in treatment for substance use disorders, and they viewed photographs of their own baby smiling and an unfamiliar baby smiling. So if you remember a few slides back, I showed you that there were specific areas implicated in reward processing, so like the striatum and the frontal cortex, that were specifically and more strongly activated when mothers looked at their own baby smiling compared to an unknown baby smiling. But in this particular study of mothers with addiction, they actually found the opposite, that they found a decreased activation when looking at their own baby smiling as compared to an unknown baby smiling in these, again, these key areas of the brain important to reward processing, like the striatum, prefrontal cortex, but also in the hypothalamus that we know is important for affiliative behavior more generally too. We've also seen that when mothers view photographs of infant distress expressions, so both their own baby crying, but also an unfamiliar baby crying, that there's an increased response in mothers without substance use disorders, but a decrease in response in mothers with substance use disorders. Again, suggesting that addiction may be attenuating the reward drive here to approach caregiving for the child. And finally, some data from ERPs. This was a study where we had maternal smokers and non-smokers view photographs of infant faces. And we looked at this ERP called the N170, that is a marker of early structural processing of faces. And we found that this N170 marker was significantly delayed in our maternal tobacco smokers as compared to our non-smokers, and that it actually was correlating at trend level with the degree of nicotine dependence that these mothers were reporting. So importantly, maternal addiction not only seems to decrease neural responses to infant cues, but also seems to delay neural responses as well. And finally, then I just wanted to comment a little bit on maternal anxiety. It's something that is generally understudied in the literature more generally, and we think is actually probably underreported. It's highly comorbid with maternal depression, but there are a number of clinical examples of where mothers are high in anxiety, but low in depression. And so it's something that we really should be paying a lot more attention to here. But we were just very curious to see what associations there may be between maternal anxiety and these neural responses with infant faces. So again, 2 ERP studies to show you. So our first study was just to look at this postnatally, to look at mothers within the first year of life. And we found that maternal anxiety correlated with neural responses to infant faces. So higher levels of anxiety, greater response to infant faces, particularly for an ERP involved in how we process attention towards infant faces. But we were curious then to think about, given our ideas around the perinatal origins of parenting and markers that might be there earlier than birth, we replicated and extended the study in a group of women in their 3rd trimester of pregnancy. And we showed exactly the same finding, that high levels of maternal anxiety were also associated with these larger neural responses to infant faces. So this to us was particularly exciting, that we could think about potentially identifying mothers who may be at risk in pregnancy using our neural markers before or not having to wait then for baby to arrive postnatally to be capturing where anxiety might be impacting processing of infant cues and subsequent responding to infant signals of affect. I wanted to then think about with you, can we then use parenting interventions to look at potential changes in maternal brain? So if what I'm saying and trying to convince you, I hope, of the value of using neuroimaging to study the maternal brain is in terms of identifying mechanisms and targets for intervention, what do we actually know about this? There's been a couple of studies now that have asked a specific question, both in parents who are low risk, but also in parents who are more high risk. So one study focused on ERPs generated by infant signals or child faces specifically, pre and post a video feedback intervention, and showed modulation of ERP specifically at the N170 that I shared with you just now in response to the parenting intervention. And then another study using fMRI, using an attachment-based intervention called Mum Power, also evidenced not only changes in neural response to infant cries, but also changes in functional connectivity in the maternal brain too, in response to the specific parenting intervention. So this does seem to suggest that we can use our neuroimaging techniques to look at the impact of interventions pre and post, but the importance will then be how we can leverage our interventions to capitalize on that knowledge in going forward. And finally, I just wanted to say a little bit about dads, as Pascal alluded to in his questions for Johnny, that we know far less about fathers, that they're incredibly understudied. What we do know, and not surprisingly, is that there's both similarities as well as differences in how the maternal and the paternal brain responds to infant faces and cries. But I think what's really interesting about studying fathers is thinking about the potential for plasticity postnatally. The dads aren't going through the same neuronal shifts or hormonal shifts specifically that mothers are going through, but there's a huge amount of exposure and experience they get postnatally. So this was one of the studies that came from Ruth Feldman's group that really demonstrated this point nicely. And so what you're looking at here in panel A is the level of connectivity between two areas that have been implicated in the parental brain. So the STS, the superior temporal sulcus, and the amygdala. And so this study was interested in looking at the functional connectivity between these two areas as a function of how much time dads were spending with their kids postnatally. So what you can see here in panel B is that the functional connectivity, or the strength of the connections between those two areas, was correlated with the amount of hours that dads were spending with their kids postnatally. So greater connectivity was associated with more time spent on average with the children postnatally. So it suggests that for dads, there may be evidence of experiential neuroplasticity as a function of experience and exposure to the infant postnatally. And certainly there will be some of that in mothers too, but at least in terms of thinking about these parental brain networks, that there is a significant role here for experience and exposure to the child that might be driving brain changes in a way that we hadn't thought about up until this point. And so just to mention then quickly some work that we've been doing looking at expectant dads and expectant mums. So this was a study where we had mothers and fathers in the third trimester of pregnancy look at photographs of infant faces, and we looked at an ERP component called the P300 that indexes how much attention we pay towards infant faces. So in expectant mothers on the left, you can see that the P300 was comparable for distress and neutral faces, but in expectant dads on the right, you can see that the P300 was significantly elevated when looking at infant distress faces compared to infant neutral faces. So again, that's really exciting for us when we think about postnatally, there's this experience piece, but prenatally, there's something else happening here, that we're starting to see this emergence of sex differences in how parents are responding to these infant cues, again, even before baby arrives. So to wrap up then, I hope I've given you some evidence to suggest that yes, there's plasticity in the human maternal brain, as much as we've heard from Johnny that's there in our preclinical studies. But really we can use and leverage our knowledge of and understanding of parental brain processes and mechanisms to allow for the identifications of targets for interventions, but also to utilize this knowledge to think about how we might look at outcomes for interventions too for parents. That we see that there are associations between maternal brain and behavior emerging, so we can take this information above and beyond what we can look at cross-sectionally in terms of thinking about longitudinal studies and the goal to predict. But critically, there's a lot of work that we need to do. So I've only given you a snapshot of what we know, but we still have so much more work to do in this area. So I'll finish there, and thank you for your attention, and thank you, Pascal, for the invitation again to be here today, and excited for your questions and comments and for the discussion too.
Brilliant. Thank you, Helena. Wonderful talk, and the consistency across the two talks is really striking. So It's you may have conferred, but nevertheless, it's just fantastic to see the commonalities there. So we're running a little bit short of time, but we've got some minutes for some questions. So please do ask your questions in the last few remaining minutes. I have some questions myself, but I don't want to hog them all. I suppose one question I had, just to get the ball rolling, is, I mean, I know I'm sort of deliberately choosing a question here that hopefully spans the two talks, which is I had a nice conversation with a very eminent ethologist some years ago, when I had started asking about, well, how do we understand and study parental behavior in non-humans, and he said you have to understand the ecology of the behavior that you're trying to study. That's the kind of the key route into understanding the underpinning neural circuits as well. So I'd invite you both to sort of think about, how do we understand the functional significance of some of these differences in parental behavior? Because obviously a key question is, how do we understand individual variation, and particularly clinically at-risk status? And I think quite a bit in what Helena's talk was suggesting, at least to me, was that there are functional explanations for some of these changes, for that could be important. But how do we kind of tie up the functional ecology of parenting with the behavior and the brain? Simple little question like that.
I can start if you want, or Johnny, did you want to go? Or...
Oh no, please go ahead.
So I think I would start off just by trying to get at what the phenomenology of the parenting scenario that you're trying to understand. I don't think that all parents parent equally, and I don't think necessarily that that we can generalize beyond just looking at how parents respond to a photograph of a distressed infant face, or listening to a specific instance of an infant cry. That I like Johnny's point earlier about how physiological states impact affiliative behavior. I think that's just as important in human parents as it is in non-human parents. But thinking about the functional implications of this is, I think, where we always kind of get a little bit stuck. To be completely transparent, is that it's all kind of well and true seeing what these neural correlates are, but it's then grounding that in terms of what their functional significance is. So that's why I think it's really important to be looking at associations between brain and behavior, so that we can start trying to really look at the manifestation of these neural circuits and what they might be for parenting behavior, but specific scenarios in which parents find themselves in. So not only in terms of the individual difference parent by parent, but also unique parenting contexts in that way too. So I think that's how I would start the ball rolling in terms of thinking about this. But I'll hand over to Johnny and see what his additions might be.
I don't have an answer. I just have maybe some observations, or some of the things that are on our minds a lot when we study, I guess, parenting in rodents. I guess it's pretty clear that the rodents that we use in the lab are quite different from the rodents that live outside the lab, and they all have a long history of domestication, during which certain traits were selected for, and especially being good parents is a trait that was highly desirable, and especially also a low maternal aggression. And that's something that we struggle with. There are certain behaviors in mothers that are simply very hard to elicit, because they have been selectively bred out of these populations. And I guess that's why I guess in our work it's important to keep in mind that natural populations, they are very different in terms of their parental behavior than the ones that we study. They have communal breeding schemes. There doesn't seem to be a lot of recognition of who's your own versus someone else's offspring. They share a lot of these duties, and that's why you also have this timekeeping mechanisms for dads. You don't rather than recognizing your own offspring, you just keep track of when you had sex last time. And all of the pups emerging 3 weeks after the time are very likely to be your own. So that mechanism makes more sense in such a communal breeding scheme, where there's a lot of alloparental behavior going on as well. But so that's important for us to keep in mind, that we have, in a way, somewhat artificial populations, and then there's a lot of effort has gone into making these populations, and that affects also the conclusions that we can draw from these animals. There's a lot of social constructs, when it comes to male typical behaviors and female typical behaviors. So we have to be very careful and always question what these animals actually do in the wild. Yeah.
Great, thanks, Johnny. And presumably some of that variability that's been bred out actually in the future could actually be very useful for understanding some of the, for example, parental aggression, which, as you say, is difficult currently. It's also quite striking that the complexity of the behaviour that's very hard to simulate in a scanner, your mice were doing beautifully, and you're seeing live, complex, ecologically based behaviour. So there's a really nice opportunity there for looking at both models complementing each other. We don't have very long, so people are very welcome to leave because we've hit 1:15 already and slightly beyond. But if anyone wants to hang around to carry on chatting, you're very welcome to. I imagine we can carry on for a couple more minutes if there are any other questions. One that came up in the chat was about the impact of parental trauma on the neural systems of parenting. And Helena or Johnny, I don't know whether you have any comments on that.
So I think that that's an area that is starting to grow in terms of its interest in understanding what trauma effects are. And so there's been some speculation just initially when looking at the impact of attachment security on maternal brain responses to infant cues, looking at secure versus insecure attachment and seeing how that impacts neuroprocessing of infant cues. There were some interpretations around maybe this is a consequence of trauma. And then Sohei Kim, who's at UMass, she started to really try and leverage our understanding of the impact of unresolved trauma and its impact on processing infant cues as well, using adult attachment interviews in that way too. So that work is starting, but it's very early on. But in terms of thinking about how mothers may be bringing trauma or prior trauma to their caregiving experiences may be impacting these neural networks as much as it may be impacting what we observe behaviorally too in interactions between them and their children.
Great. Thanks, Helena. I suppose I'm aware of lots of interesting studies on rodent models of early life adversity. And so there are models for that, which could also then relate to parenting in rodents too, which would presumably give you quite a nice model. I imagine people have done that sort of work. Is that right, Johnny?
Yeah, there's a lot of work in rats, I guess, tracking the later behavioral outcomes of your own upbringing, suggesting that if you were exposed to good parenting, you're more likely to be a good parent later on. There's a lot of really amazing work happening there.
Yeah. Well, I'm feeling guilty that we ought to stop really because we said we'd finish at 1:15. I wanted to say, can we do this again sometime? Because it's been incredibly thought-provoking and really exciting to hear both of your talks. Thank you so much for your time. I hope that it's inspired the UCL and wider community to think about the neuroscience of parenting, which is a very under-neglected subject, in fact, compared to lots of other topics in neuroscience, and they're crucial for understanding child well-being. So thanks very much for coming, everybody, and look forward to seeing you at the next Catalyst seminar.
Thank you to both speakers who were just brilliant. There's lots of thank yous. I think the speakers should look at the chat there, a huge amount of thank yous. And I think we probably didn't manage all the questions, Pascal, so I think people could be encouraged to drop an email if there is something that's outstanding following the talks. I'm sure that the speakers...
Great. Yes, if you wouldn't mind, that would be wonderful. Great.